Hello, my name is Jefferson Stepp, and I will be doing human papillomavirus. What is HPV? HPV is a sexually transmitted infection with more than 42 million Americans that are currently affected. There's almost about 13 million that become infected every year, and there's over 100 plus different types of HPV. There are also low-risk types and high-risk types. Some examples of high-risk types would be HPV 16 and HPV 18. An example of some low-risk types would be HPV 6, HPV 11, HPV 42, HPV 43, and HPV 44. Cervical physiology. So the cervix is a very important part of the female reproductive system. It also keeps bacteria out of your uterus and produces discharge to clean your vagina. It changes its position to help facilitate sperm or protect a pregnancy from even happening. So there are two main cell types. Squamous cells are flat cells that look like fish scales under a microscope. These cells are found in the tissues that form on the surface of skin to help protect from intrusion while also being able to use selective diffusion to allow materials to pass through. Adenocarcinoma cells are cells found inside the lining of the organs, such as the cervix, helping to produce and secrete bodily fluids, including mucus, and they can be found in the inner part of the cervix and uterus. Cells and tissues targeted by human papillomavirus. Epithelial cells perform a variety of functions that include protection, secretion, absorption, excretion, filtration, diffusion, and sensory reception. HPV will infect and replicate in squamous epithelium only. And the cervical intraepithelial neoplasia can be found by a Pap test. It is most often caused by HPV, and there are different stages that categorize the CIN. CIN1 invokes about 1/3 of the thickness of the epithelium. CIN2 refers to the abnormal changes in about 1/3 to 2/3 of the epithelial layer. CIN3, which is the most severe form, affects more than 2/3 of the epithelium. E6 and E7 oncoproteins with HPV 16. Oncoproteins are a protein encoded by an oncogene, which can cause the transformation of a cell into a tumor if introduced into it. A prime example of this would be the E6 and E7 oncoproteins. Apoptosis is the death of the cells, which occurs as a normal and controlled part of an organism's growth or development, which can be blocked by the E6. The E6 oncoprotein is also responsible for activating the telomerase, which is responsible for the immortalization of primary keratinocytes. E6 oncoproteins and this activation is mediated by part of the E6 induction of the H-Tourette promoter. E7 oncoproteins and HPV. The PRB is responsible for blocking tumors, and the E7 binds and inactivates the PRB suppressor protein. E7 is also responsible for reprogramming the cellular environment to be conducive to viral replication, which makes it easy for the virus to spread. This can be shown below with the first figure. The E7 attaches to the RB and is responsible for blocking it. E6 oncoproteins and HPV 16. The P53 protein primary role is to regulate the cell division and death, and when it's infected, it can cause the cancer to spread even more. E6 protein inhibits apoptosis by targeting different proteins, including the P53, and it causes degradation. E6 also protects cells from death receptor-induced apoptosis by blocking apoptotic signal. Degradation of P53 causes the epithelial cells to be attacked even more through malignant transformations, which, if not controlled or treated, could worsen the cancer. How does E6 and E7 oncoproteins tie into HPV 16? HPV encodes two oncoproteins, E6 and E7, which are directly responsible for the development of HPV 16, which is a high-risk cervical cancer. E6 degradation interferes with the biological functions of P53, which makes it easy for the cancer to control the cell cycle progression which leads to increased tumor growth without the regulation of killing the bad cells. E7 PRB is responsible for blocking tumors, and the E7 binds and inactivates the PRB. Diagnosis of HPV. A pap smear is a procedure with a small brush which is used to gently remove the cells from the surface of the cervix and the area around it so it can be checked under a microscope for cervical cancer. CIN stages are used to see how bad the cancer is and how deep into the epithelial cell it is. A colonoscopy is a biopsy method used to open up the vagina and see what's inside while it being held open. This is usually painful and is a long procedure. Treatment options for HPV. During chemotherapy, cisplatin is the main drug used to kill the cancerous cell. There are surgical procedures such as cold knife cone, which is a surgical procedure used to treat cervical dysplasia or early cervical cancer. It involves the excision of a cone-shaped portion of the cervix to remove a cervical lesion along with the entire transformation zone. In a total hysterectomy, the womb and the cervix are removed through an incision that is made at the top of the vagina. After the womb and cervix have been removed, the incision is sewn up. Radiation therapy uses high-energy X-rays to kill cancerous cells. Depending on the stage of the cervical cancer, radiation therapy may have to occur more than once and could take a while. Treatment timeline for HPV. It takes most women around 2 years to clear up the condition, but depending on what stage they are in, could make it longer. There is no medication that can treat HPV itself, but there is treatment available for the side effects of HPV.